Today we're going to be discussing Chapter 2, which I call the chemical context of life. Uh, Double check your reading assignments. Not very long, much reading today. Maybe it's a bit of review for you if you've had chemistry in the past. A good refresher, trying to put chemistry into context in terms of biology, but also in terms of healthcare and your future careers. And I'd like to start this discussion with a quote from a gentleman named Paracelsus. He lived in the late 1400s, early 1500s. He was a Swiss alchemist, but he also was a practitioner in medicine at the time. And he said the art of healing comes from nature, not from the physician. Therefore, a physician must start from nature with an open mind. And that is one of the most true sentences when it comes to healthcare. What we're trying to do in healthcare, and in a larger sense, because many of you are planning to go into healthcare, is that you have to understand what we're trying to do is we're trying to manipulate the system in order to achieve balance or normality in the system. So we have to understand that system, and the system of biology is chemistry. The fabric of biology is chemistry. So I want to go over a little bit about some of the basics of chemistry that we're going to kind of keep reinforcing over the semester. And I include this slide right here, not because it's testable, but the fact that here's how chemistry can affect us in our lives. Certainly we talk about ecosystems and chemicals in the environment, our lifestyle, the types of foods and exercise that we get, the types of habits we have, the interactions we have with others, as well as the types of materials we might use for healthcare. There are all sorts of interactions. Like I said, it's the fabric of our lives is chemistry. Just like the fact that air and an aircraft work together to create flight. Air is not only on which the wings glide over, but air is also the fuel in those engines. That's the way chemistry is in biology. We have to understand a little bit of chemistry just to make sense of what we're seeing in the biological systems. The first thing that we're going to do is talk about atoms. Why? Because the electron serves a critical role. There are actually 4 major roles of electrons in biological systems. But we're also going to re-emphasize the idea of structure and function. Structure and function is certainly true when it comes to biology, when it comes to physiology, but it's also true at other levels. The structure of the atoms and even the subatomic particles gives rise to their function in systems. The structure of molecules, the structure of cells and tissues and organisms and even societies all dictate their function. So it's a scalable concept. And so we're going to break up the atom a little bit, and we're really going to focus on the electrons mostly. Each of these different elements here, elements are concentrations of a given type of atom, a different structure of atom. We have a common list of elements that we find in the human body. These are the most common. We certainly have others, like for instance iodine, iron, manganese, selenium, zinc. These are all what we call trace elements, where these ones up here, particularly these 5 up here, we find in great abundance. In fact, 96.3% on average of the mass in the body, including the water, is made up of just these 5 atoms. The other almost 4% is found in these, particularly calcium, right? Phosphorus, that's because of the bones, all right? And then we have a bunch of trace elements down here. But each of these elements differ in the atomic structure and their relative chemical reactivity, which comes back to our electrons. So we have atoms, and the different number of protons that we find in the atom dictates what type of element that it is. Also influences what number of electrons that we have surrounding the nucleus of an atom. If we have a certain number of protons and neutrons, this atom may be actually unstable and be what we call radioactive. Radioactivity has to do with the fact that the nucleus of an atom is unstable, and so parts of that nucleus will be given off. And we have different kinds of radiation. But that's all I want to say for now. The number of protons dictates, in part, the number of electrons surrounding that nucleus, and thus will confer function. Some atoms are exceptionally non-reactive, things like helium and argon, and some atoms are incredibly reactive, so reactive that if you threw them into a tub of water, they would explode and be quite dangerous. So atoms can be a combination of either, they can be unreactive or they can be incredibly reactive, and it has to do with the structure. The structure dictates what that atom's going to do in the environment. When it comes to biology, most of the action that we're talking about is at the level of the electron. And electrons serve multiple important roles in biologic systems, one of which is as an energy acceptor and an energy donor. Well, energy is the ability or the capacity to cause change, particularly change in motion. And so when we impart energy on something like a basketball, we can push it against gravity. And then when gravity takes over, that ball will fall down. And so we have energy flow going on in this system because the motion is changing. We also have energy in the form of motion at the atomic and the molecular level. That's things like heat, for instance. Things like fluorescence, when we shine a UV light on something and it glows a certain color. That's energy that's flowing through that system, and that's a molecular energy situation. But it's electrons absorbing and then releasing energy. We also have potential energy and kinetic energy. So there's potential energy because of its location or structure that then gets turned into motion. That's kinetic energy. So kinetic energy is the energy of motion. For instance, if you have more heat, you have more kinetic energy. We will break chemical bonds in order to create heat, or heat is created as a byproduct of breaking those chemical bonds. So the chemical bond would be potential energy, and the heat arising from breaking that chemical bond would be kinetic energy. So electrons absorb and release energy, and that can cause phenomena to occur in those electrons, either the forming and breaking of chemical bonds, the fluorescence that I mentioned about, and other roles that I'm not prepared to talk about today. That's more for physiology, but in any case, that's what makes electrons so very, very important in biologic systems, that they can switch from potential and kinetic energy in a number of different ways, which include chemical bonds. And that's what I really want to talk about. And here are some common molecules that we find in biologic systems, like hydrogen gas and oxygen, water, and methane, one of the most common organic compounds. What do we see here? What kinds of chemical bonds are these? We see this shared electron situation. Those make these covalent bonds. Are they strong or weak? That's one of the key questions. And are these bonds polar or nonpolar? All of these questions are ones that you're going to want to be able to answer when you look at a molecule or a molecular structure, because the idea of understanding how that bond will behave, whether it's in water or it's not in water, how strong or weak it is, will affect whether or not it will hold up to certain types of stimulus or strain. And polar or nonpolar, it kind of gets back to the idea of water. Because we are aqueous organisms, because life requires water, understanding polar and nonpolar becomes incredibly important. For instance, if we look at this right here, we would say that it is nonpolar because we have two oxygens and they pull equally on the electrons, and so these bonds are nonpolar. Okay, Whereas these bonds right here, the oxygen pulls a little bit harder on the electrons than the hydrogens do. And so even though these are covalent bonds, they have a little bit of polarity. There's a little more negative charge on this side versus this side. We're going to talk about that a little bit more when we actually get into the chapter on water. So when it comes to covalent bonds, which are very, very important, and again, we'll get to ionic and other bonds later on, especially when we get to proteins. We have nonpolar covalent bonds. These ones are where the electron is equally shared across both atoms. The atoms do not have to be identical, or the areas in which the atom or the molecule do not have to be identical. In a diatomic element where we have two of the same, that's an example where you have the same atom on either side. And so that by definition has to be nonpolar because each side is pulling on that electron equally. We call that pulling force on that electron electronegativity. The stronger the electronegativity, the more that that atom or group of atoms pulls on those electrons. So the less electronegativity, the more likely that the atom or the group of atoms is going to let the other guy have more of a say about where that electron is. So nonpolar, we have equal electronegativity, and so that those electrons will spend as much time over here as they will over here. They're not standing still. They're actually in orbitals, and they're in constant motion. But the probability that they're going to be over here or over here is equal. That changes, though, when you have differing electronegativity. When you have differing electronegativity, the frequency of time that this electron is going to be on one side or the other changes. It's no longer 50-50. The atoms or the groups of atoms do not share the electron equally. And like for instance here, this is actually so electronegative that it's almost ionic. It's basically an ionic compound. So ionic compounds have enormous difference in electronegativity. Other compounds, though, that are kind of like ionic bonds, but not quite, have differing electronegativity. And so the electrons may be shared, but they're shared unevenly, if you will. Water is another great example of this, where we see uneven sharing of the electrons, and that's why water is a polar compound. And so understanding polar and nonpolar helps you understand whether something is water soluble. Most polar compounds are water soluble. Most nonpolar compounds are water insoluble, or what we call hydrophobic. They tend to be more oily. They tend to be less able to dissolve in water. That's where all of this goes. Like I said in the previous slide, because of differing electronegativity between the oxygen and the two hydrogens, we have the electrons spend more time over on the oxygen side. And that gives this side of the molecule a little more negative charge. So we have this little funny looking symbol. That's a Greek letter delta and a negative next to it. Whereas on the hydrogen side, we have the delta with a positive. And so water is a polar compound. You can do this experiment at home. If you have a thin stream of water coming out of a faucet or something, you can hold a magnet, and maybe you can find a good strong magnet. You can actually pull that water over because the water has magnetic properties due to the fact that it's unbalanced in its charge. It's got a negative and a positive side. Magnets have negative and positive sides too. When we have electronegativity that is so great and so little in two elements that are reacting together, we can have where the electron is basically spending all of its time on one element and hardly any of its time on the other. This leads to something that we call electrostatic attraction. And the electrostatic attraction, the +2 versus the -2, gives us what we call an ionic bond, because the electron is spending so much time over here, it's virtually not spending any time. That means that these things are now ionized. They have a durable electrical charge that does not go away. Even if we were to break these two atoms apart, they would generally retain the electron, or not retain the electron and remain as ions. Now, are these bonds strong or weak? I'd like you to read a little bit more about that in your book. Are these polar or non-polar? Well, you can see that we have a +2 and a -2. That's a fairly self-evident answer there. And in what chemical compound do we often see these bonds located? So make sure that you're answering these questions, that you can identify ionic bonds, and you can often identify them because they'll have a full-on charge. You don't see that delta symbol anymore. You see whole numbers. The delta symbol is a positive, or a partial positive, or a partial negative. Here we have whole numbers, which means the equivalent of an entire electron. Two more quick terms for you. When we have a neutral atom that has either gained or lost an electron, like in our ionic bond, we have a cation, positively charged. You can remember cation because the T looks like the plus. And then we have anion, a negative ion. Okay, that makes it easy to remember which is their cations and anions. And once they form these, then they have that electrostatic attraction that will allow them to hold together. In solution, though, they can disassociate, particularly in polar solvents like water. This figure here is showing you two different types of chemical bonds, and we have the solid lines, which indicate covalent bonds. These are polar covalent bonds, and the reason you know that they're polar covalent is because we have a delta negative on this atom, and we have delta positives on these two atoms. So these are polar covalent bonds. That's a water molecule, and there's another one here that's tilted on an angle, and another one here, and so forth. But we also seem to have these interactions with these dotted lines. That implies some type of chemical interaction going on, a chemical bond, if you will. So what are these bonds? These are known as hydrogen bonds. Hydrogen bonds always have hydrogen, first of all. They always have hydrogen, and then they have one of two other elements. So here's our covalent bond as delineated with a solid line, and hydrogen bonds are delineated with this dashed line or dotted line. What three atoms are the only atoms potentially found in these bonds? Well, we can see two of the three here. We always have hydrogen. It's a hydrogen and oxygen. So that's two of the three. The other atom is nitrogen. And where we're going to see hydrogen bonds and nitrogen is when we look at the DNA structure. We also see it in proteins, but we'll really see it in DNA structure. So if you see those dotted lines, that implies a hydrogen bond. It's going to either be between a hydrogen and an oxygen, or a hydrogen and a nitrogen. But that oxygen and that nitrogen have to have a partial negative charge. They will not just bond with water spontaneously. They have to have that partial negative charge. For instance, you know that fish can swim around in water and they have gills and they can absorb oxygen from water. They're not absorbing this oxygen. Okay, they're not absorbing this oxygen. The oxygen that we have there is actually dissolved in between the water molecules. So it's not reacting. Those oxygen molecules aren't reacting in the same way that this hydrogen bond is. Is it strong or weak? It's relatively weak. This is a relatively weak interaction. Doesn't mean it's not somewhat strong. It's not the weakest, but it's generally less weak than ionic, or more weak than ionic, and generally a lot more weak than covalent. So out of the four bonds that we're talking about today, it ranks 3. Now in this figure, we actually have our covalent bonds that we see with that solid line. They're inferring some covalent structure here with this blue, but we won't worry about that too much. But these solid lines, those are covalent bonds. Over here on the right, we can actually see an ionic bond in this protein. We have a plus and a minus. This electrostatic interaction is causing an ionic bond to form between these two side chains of the amino acid. Over here with A and B, amino acid A and amino acid B, we have those dotted lines again. Okay, so this is a double covalent bond, but these dotted lines imply a hydrogen bond between hydrogen and this delta negative oxygen right here. But what's going on here at C? What's going on here at C is known as a van der Waals interaction. We also can call them hydrophobic interactions. To make a long story short, if we look at this side chain here, what we see are lots of C's and H's. Lots of C's and H's. So what we have is a cloud of electrons. These carbons all have a relatively similar electronegativity, and so what happens is the electrons kind of form a sort of a shared cloud around there. This is why we study organic chemistry a little bit. Organic chemistry is really about how electrons flow around molecules. But in any case, this is a very nonpolar, a very hydrophobic side chain of an amino acid, and we actually have a couple others right here. Those are going to have those clouds of electrons as well. Well, those clouds of electrons can cause a hydrophobic interaction that we call a van der Waals interaction, and it's the weakest of the chemical bonds. The strongest is the covalent, distantly followed by ionic, then hydrogen, and then lastly these hydrophobic interactions we call van der Waals. Doesn't mean they're not important. Even though they're weak, what they're going to do is they're going to cause hydrophobic side chains of amino acids to want to glom together and create hydrophobic regions inside the protein. But that's for another day. Right now we just want to understand the bonds, and so we have covalent that we see here in these solid lines right here. There's a double covalent bond here. We have ionic, and you can always see ionic because they'll have the positive and the negative there. You don't see a delta. There's no delta. We have our hydrogen, most often demonstrated without the deltas and a dotted line like so. And then lastly, it's hard to identify these except that you would see lots of C's and H's. These would be very, very hydrocarbon-like side chains. That would be a hydrophobic, otherwise known as a van der Waals interaction. So the structure of the atom and the reactivity of the atom, the energy state of the electrons and the bonds they form, give rise in part to what causes molecular shape and thus function. A molecule's shape is usually exceptionally important to its function, and there's no more evident situation than when we have two signaling molecules in the human body. One is called estradiol and the other is called testosterone, the Mars versus Venus. These different molecules give rise to exceptionally significant changes between males and females, not only structurally, but in terms of cerebral function and so forth. So structure and function leads to the outcomes that can be vastly different in different individuals. So we determine the shape of the molecule by the positions of the atoms, the bonds that they form, the energy that is in those atoms, and the electron states. Another example of this, these are actually identical compounds in terms of their molecular structure, but one is left-handed and one is right-handed. The left-handed one's a very, very potent opioid peptide called morphine, and the one on the right is a much less potent form of opioid called dextromethorphan. It's useful as a cough suppressant. Morphine can also act as a cough suppressant, but it also acts in other ways and is also highly addictive. Dextromethorphan is somewhat addictive, but not nearly so, and that's because of its arrangement of its atoms. Even though it's almost identical, it's a mirror image, and that means that it's not nearly as biologically active. And here we have a picture from your book talking about how morphine does its thing. That's that left-handed molecule we saw a second ago. It mimics, you can see the structure right here, it mimics the shape of a natural peptide or set of peptides that we find in the human body that we call opioid peptides. So morphine here is acting as a mimetic in this situation. That's a pharmacologic term. And the final concept I'd like to introduce to you today is the idea of chemical equilibrium. All chemical reactions, whether we're talking about biological ones or non-biological ones, are reversible, assuming that we can get the appropriate conditions. It might require an awful lot of energy, high concentrations, and so forth. But in biologic systems, all chemical reactions are generally reversible in relatively narrow window of conditions. It's actually reversibility is actually quite common. And so we have our reaction here, A plus B, leading to a product, if you will. So the reactants on the left and the product or products on the right. In all biological reactions generally obey this idea that they can move in both directions, assuming the conditions are met. And usually those conditions are pretty subtle compared to non-biologic reactions. This idea of reversibility and motion back and forth is known as chemical equilibrium. That is to say that we are going to see, A plus B reacting to form AB, and we're going to see AB disassociating to form A plus B, and that's why we have arrows in both directions. In aqueous situations where we're dissolved in water, this is much more the norm, where we have a certain percentage AB or A plus B and a certain percentage bound AB. So this is a very, very common situation in biology. We talk about hormones with their receptor or drugs binding receptors in the body. Like I said, all chemical reactions are reversible, even very, very durable molecules. This is an image right here of a diamond. This is an actual diamond that is burning. If you give it enough heat and you drop it into liquid oxygen, the carbon in the diamond will form carbon dioxide. But it requires very, very extreme conditions because those covalent bonds are very, very stable. That's why diamonds can exist for billions of years without decaying. But if we get enough heat in there and enough oxygen, we can create carbon dioxide. But back to our more normal situation in biology where we have things associating and disassociating. Very, very important in pharmacology. We design a drug, we want that drug to bind a receptor to cause an effect. If that drug is not very potent, it may not bind very well to that receptor. It may still bind, it's just not very much of it's going to be bound. And that's the point I want to make, is that when we reach equilibrium, the rate of the reaction going this way is the same as the rate of the reaction going this way. And our bodies are going to use that desire to reach equilibrium. If we have lots and lots of A plus B, it's going to want to form AB. Well, if we start controlling the amount of A plus B, we start controlling the energy in the system, we can control whether or not it forms AB. If we have a high amount of AB, we might start to see it disassociating into A plus B. This interaction is a little more complex than your normal chemical reaction, but this becomes very, very important, especially when you move on into physiology and so forth. But I want you to understand chemical equilibrium. That's when the rate of product formation is the same as when the rate of product breakdown, when they're identical. They may not have the same amount of mass on each side, but they are moving at the same rate in both directions, and that's chemical equilibrium. So to summarize today, we went over a little bit about the atom with particular attention to the electrons because of their role in the ability to absorb and release energy quite readily. We did talk a little bit about the common elements. You should be able to recognize the common elements, those top ones. the structure-function relationship not only occurs at the atomic level, but also at the molecular level, and we'll see that relationship again and again and again. In this case, we emphasize those energy levels in the electrons. That forming chemical bonds, the types of bonds, which ones are strongest, which ones are weakest. What are the four bonds that we talked about? Which ones are polar and which ones are nonpolar? We talked about ions, the idea that we have ions that are where the number of protons is not the same as the number of the electrons, and how those ions contribute to bonding. And then lastly, the structure-function relationship with molecules and that idea of chemical equilibrium. So those are the highlights today. Please do just read those few pages that I assigned to you. If you feel weak in any of these areas, we'll try to reinforce some of this during our synchronous time. So thanks for your time. We're right at half an hour, and I will talk to you soon.